Okay.
Cool. Thank you, Kenny. All right, so maybe we'll just get going. Welcome to the bioengineering seminar, first one in the summertime. And we are so excited to welcome Dr. Miki Ebisuya from European Molecular Biology Lab in Barcelona. She did a PhD in Kyoto, Japan, probably fair to say the mecca of regenerative medicine in Japan, but as well as probably in the world as well, one of the meccas. And she stayed on there to start her group quite soon after PhD. So you must have done really nice job in PhD. And then she moved her group to RIKEN in 2013. And then the group moved to Barcelona in 2018, and we've been hearing quite a bit about her exciting work in her from her group, and it's really wonderful that we can listen to some of it today. So I have this habit of asking speakers I host that something about them that's not on their CV. So I asked Miki what was her lockdown hobby, and she kindly told me that. So her lockdown hobby, one of them was audiobook. So she started listening to audiobook in the past year, I guess. And so these days, every day she walks to work for 40 minutes each way, and while listening to audiobook. And currently, she said she's listening to Kazuo Ishiguro's Clara and the Sun. Thanks very much, Miki, for being here today, and please take it away.
Thank you, Naomi, for inviting me. And yeah, the book Clara and the Sun is a very good one. So if you are interested, please take a look. So let me share the screen. Sorry, but not this one. Sorry, something is not working quite right, but can you see my monitor? Yep, I can't see you, but if there's any problem, please just speak up. So hi everyone, I'm Miki Ebisuya from EMBL Barcelona. So my lab works on synthetic developmental biology. We combine synthetic biology and developmental biology. So basically, we recapitulate developmental processes by using pluripotent stem cells. And then we use those in vitro models for 3 different purposes. The first purpose is to compare different species in vitro. And the second one is to study congenital diseases by using those in vitro models. And the third purpose is to develop several tools to manipulate organoids and in vitro models. So today I want to talk mainly about the first one, cross-species comparison. And since this one is more like a developmental biology project, and if I still have time, I will probably touch upon the manipulation part. And this is more like a synthetic biology project, which may be more suitable for this audience. So anyway, in the cross-species comparison part, I'm going to talk about why human time tends to be longer, slower than mouse time by using in vitro models. So this particular project, human time, mouse time project, started almost 20 years ago when I was a high school student. I read a Japanese book titled Elephant Time, Mouse Time, written by Tatsuo Motokawa. So this very interesting book taught me that different animal species live with different time scales. For instance, the gestation period of humans is 9 months, but it's just 20 days for mice, but it's almost 2 years for elephants. And we larger animals tend to live longer than smaller species. And our cardiac cycle, the heart beating rate, is much slower than that of mice. So I was fascinated by these facts. So my biological time is different from their time. And if I focus on developmental time and developmental time of mice and humans, they are quite similar. For instance, day 10 mouse embryo is quite similar to day 20, 30 human embryo. Their morphologies or even the sizes are quite similar. And on day 20, mouse babies are already born, and human babies stay in the uterus after 60 days, and then they keep growing. So after day 60, things will be completely different. But until this point, morphologies and sizes are quite similar between mice and humans. But as you see, the progression speed is slower in humans, maybe 2 to 3 times slower in humans compared with mice. And this is a question I want to address. Why is human development slower than mouse development? However, gestation period or entire development is too complex and too long for me to study. So I needed a simpler, shorter model system to compare human and mouse time. And that's why we focused on the segmentation clock. The segmentation clock is an oscillatory gene expression during development. And this movie was taken by Ryo Kageyama's lab, Kyoto University. And this is a tail bud of a mouse embryo, day 10 mouse embryo. And during this stage, this gene HES7 is known to show oscillatory gene expression. So this yellow color indicates the expression level of HES7. And if we focus on one part of a movie, let's say here, the expression level of HES7 goes up, down, up, down, up, down, up, down. So it's oscillating. And it's also a wave from tail to the head part of an embryo. And this wave and oscillation are called the segmentation clock. And this segmentation clock, this oscillation is important to determine the timing of our body segment formation. But for now, please just remember that the segmentation clock is an oscillation important for development. So in mice, this segmentation clock is well studied, but unfortunately, we can't really do time-lapse imaging with human embryos. So that's why recently we've recapitulated this segmentation clock by using human iPS cells in collaboration with Janta Shalef, Yoshihiro Yamanaka, Makoto Ikea, Junya Toguchita, and other wonderful colleagues in Kyoto. So basically, the treatment of human iPS cells with a cocktail of growth factors and inhibitors can convert human iPS cells to human presomatic mesoderm cells, or PSM cells. Those presomatic mesoderm cells are the cells that show the oscillatory gene expression during early development. And then Mitsuhiro Matsuda, a postdoc in my group, tested whether those induced PSM cells can show the segmentation clock oscillations. So he made an HES7 promoter luciferase reporter and then introduced it into human iPS cells and then induced the PSM cell fate. So in this movie, we are probably looking at several hundreds of cells. And we are looking at the expression level of HES7, which goes up, down, up, down, up, down, up, down, up, down. So it's oscillating. The expression level of H7 is oscillating in induced presomatic mesoderm cells. So we can recapitulate the oscillation of the segmentation clock. But not only the oscillations, but also the traveling waves of the segmentation clock we can recapitulate by using human iPS cells. So in this case, we made a spheroid, an aggregate of human iPS cells initially, and then treated this spheroid with a cocktail of growth factors and inhibitors, and then to induce the PSM cell fate, and then put the spheroid on a dish. So one spheroid contains about 10,000 cells. And again, we are looking at the expression level of this H7 marker gene, which showed several oscillations. As you see, pulse, 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 pulse. So we can see the oscillations. But at the same time, I hope you can see the traveling waves from right to left in this movie. Just like the waves from posterior to the anterior part of an actual mouse embryo. This way, we can recapitulate both oscillations and waves of the segmentation clock by using human iPS cells. Then our main purpose was to compare these human oscillations to mouse oscillations. This part was also done by Mitsuhiro, and Hanako Hayashi also helped the project. So very interestingly, if we use human iPS cells, human stem cells, as a starting material for in vitro segmentation clock, the oscillation period was 5 to 6 hours. I mean, one cycle of oscillation or one wave takes 5 to 6 hours in humans. But if we use mouse stem cells, mouse ES cells or mouse epiplast stem cells instead of human stem cells, then the oscillation period was 2 to 3 hours. And these values are consistent with in vivo measurements. In our body, our body segments are formed every 5 to 6 hours. And in mouse bodies, their segments are formed every 2 to 3 hours. And here's an overlay of mouse and human oscillations. And in this movie, on the left are mouse cells, mouse presomatic mesoderm cells. And on the right are human presomatic mesoderm cells. And we are looking at H7 expression again. In the mouse sample, H7 expression goes up, down, up, down, up, down, up, down, up, down quite quickly. But in the human sample, it goes up, down, up, down, up, down more slowly. So actually, we found that human oscillation is 2 to 3 times slower than that of mice. And that's a question I'd like to address here. Why is the human segmentation clock period 2 to 3 times longer than that of mice? So before addressing this question, I'd like to briefly explain the mechanism of this oscillation. It's quite simple. The core of the segmentation clock is just one gene, H7. So this H7 gene is produced through transcription or translation, and then H7 protein is degraded, just like any other gene. However, this H7 gene is a transcription replacer that inhibits its own promoter. So it's a one-factor negative feedback loop. And this negative feedback loop can give rise to an oscillatory gene expression of H7. Really simple. And we think this negative feedback loop, H7 itself, is conserved between mice and humans. The mechanism itself is conserved. But instead, we found that the production processes or degradation processes are slower in humans compared with in mice. So we found that several biochemical reactions of H7 are slower in human cells. For instance, the protein degradation of H7 was 2 times slower in human cells compared with mouse cells. And the delay due to transcription translation was 2 times longer in human cells. And even the delay due to intron splicing was 3 times longer in human cells. So let me show you a bit of actual data, actual measurements. So this is a measurement of protein degradation of H7 in both mouse cells and human cells. By the way, if I say a mouse cell, that means a mouse presomatic mesoderm cell, PSM cell, and a human cell means human presomatic mesoderm cells. The point of our study is to compare the same cell type between different species. We can't compare human skin cells to mouse neurons, for instance. So anyway, there are 4 combinations here. We measured the degradation of mouse H7 protein in mouse presomatic mesoderm cells, or human H7 protein in mouse cell, mouse H7 protein in human cell, and human H7 in human cell. But basically, both mouse and human H7 proteins, mouse and human H7 constructs were degraded much faster in mouse cells compared with in human cells. So it's not the sequence difference between mouse and human H7 genes. So they are essentially the same. But instead, we think the cellular environments where H7 function are somehow different between mice and humans. So human cells and mouse cells are somehow different, and this host cell decides the degradation rate. And for some unknown reasons, H7 degradation becomes 2 times slower in human cells. So protein degradation is 2 times slower. And similarly, we also measured the delay due to transcription or translation. Again, we used an exogenous reporter, which comprises an inducible promoter and H7 luciferase reporter. So this exogenous construct was induced much earlier in mouse cells compared with in human cells. So we defined this onset timing as a delay due to transcription translation. And such a transcription translation delay was about 2 times longer in human cells. So transcription translation is 2 times longer in human cells. I won't show the actual data, but we also found that the delay due to intron splicing is 3 times longer in human cells. So by using these measured parameters, the degradation rates and delays measured in human and mouse cells, we made a simple mathematical model in collaboration with Joldi, Gamshin, and Halbo to simulate actual oscillations. So very interestingly, if we use human parameters, the degradation rates or transcription translation delay or intron delay measured in human cells, then the simulated oscillation period was 5 to 6 hours. But if we use mouse parameters, the degradation rates or delays measured in mouse cells, then the simulated oscillation period was 2 to 3 hours. And luckily, these values are consistent with the actual human and mouse segmentation clock periods. Therefore, we concluded that human segmentation clock is 2 to 3 times slower than that of mice, because the protein degradation of H7 or transcription translation or even splicing, those biochemical processes are 2 to 3 times slower in human cells compared with mouse cells for some unknown reasons. So that was our conclusion. So biochemical reactions are slower in human cells for some unknown reasons. So obviously, what we need to address now is the mechanism. Why are those reactions slower in human cells? We are working on it, and we have some hypotheses, but we don't have answer yet, unfortunately. So that's what we need to address next. But in parallel, I'm trying to explore the universality of this mechanism. The slower biochemical reactions in human cells specific to this Hess7 gene or generalizable to some other genes? Well, so far, we've focused on mouse and human segmentation clocks. But what about other developmental processes? What about other species than mice and humans? So let me show you a bit of recent data, preliminary data, regarding the universality of the mechanism. So as for the other genes, this question, we've recently systematically measured the degradation rates, protein degradation rates, by using dynamic SILAC method. It's a protein labeling, a radioisotope labeling method in collaboration with Misha Savitsky and Henrik Hammeren's group. So those green dots in this graph indicate the each gene, and there are about 4,000 genes in this graph. And the x-axis of this graph indicates the half-lives measured in human PSM cells, presomatic mesenchymal cells, and y-axis is the half-life measured in mouse cells. And here is this y equal x black line. And compared with this black line, the distribution of green dots is shifted to the right. That there's a general trend that human cells show longer half-lives than mouse cells. Actually, among about 4,000 genes we measured, 1,000 genes showed significantly longer half-lives in human cells, while only 250 genes showed significantly shorter half-lives in human cells. So I think it's a general trend. Human cells tend to show slower half-lives, longer half-lives than mouse cells. So I would say the slower reactions in human cells are generalizable to other genes to some extent, but I wouldn't say that all genes should show this interspecies difference. It depends on the gene. Then what about other developmental processes than the segmentation clock? Recently, James Briscoe and Teresa Ryon's group studied motor neuron differentiation, human and mouse motor neuron differentiation processes, and then they reached a similar conclusion. So the motor neuron differentiation processes are quite conserved between mice and human. The sequential induction of marker genes is conserved, but so the pattern is conserved, but progression speed is again slower in humans, maybe 2 to 3 times slower in humans. And then the authors measured the protein half-life, bulk protein half-lives in human and mouse motor neuron precursor cells, finding that the half-lives are 2 to 3 times longer in human motor neuron precursor cell. So I'd say the slower biochemical reactions in human cells may be a universal mechanism that can explain the slower pace of human development compared with mouse development, even though it's too early to say this. And then what about other species than mice and humans? So these days, my lab has been collecting pluripotent stem cells, ES cells or iPS cells of several mammalian species to make a stem cell zoo in the lab. So we've obtained several stem cells, several species, including rabbit ES cells, pig iPS cells, cow horse ES cells, and even rhinoceros ES cells. And we are also working on establishment of new iPSC lines in collaboration with neighborhood institutes and European zoos. So we obtained those animal stem cells from wonderful collaborators, but those cells are quite difficult to handle, to be honest. We need to optimize the induction protocol, differentiation protocol for each species. But luckily, my lab has got talented zookeepers recently. Maria Costanzo and Jorge Razalo, the two PhD students, are taming these animal stem cells. So they've succeeded in taming rabbit and cows and rhinoceroses. So they succeeded in converting those stem cells to presomatic mesoderm cells, PSM cells. And then rabbit segmentation clock showed 2.8 hours oscillation period, which is quite similar to mouse period, maybe a bit longer than mouse period, but still quite similar to mouse period. And cows showed 4 hours oscillation periods, and even rhinos showed about 4 hours oscillation periods. To be honest, personally, I expected these larger species to show longer oscillation periods than humans, but yeah, they showed shorter periods than humans. These data are still quite preliminary, but this is our current summary table of the segmentation clock period 2. So mice and rabbits showed 2 to 3 hours oscillation period. Cows and rhinos showed 4 hours, about 4 hours periods. And so far, we humans showed the longest periods of 5 to 6 hours. So currently, we are studying on whether these differential periods are derived from differential biochemical reaction rates across species. All right. And our ultimate goal is to explain several examples of elephant time and mouse time by using in vitro models, stem cell derived models. To wrap up this part quickly, we've recapitulated the human and mouse segmentation clocks by using human and mouse stem cells. And human segmentation clock showed 5 to 6 hours, while mouse cells showed 2 to 3 hours periods. That's because the protein degradation, transcription, translation, splicing, these processes are 2 to 3 times slower in human cells compared with mouse cells for some unknown reasons. All right, that was the first part. So let me skip the second part. And with the rest of the time, I'd like to talk about manipulation of in vitro models. So this is more like a synthetic biology project, and I consider myself as a synthetic biologist. I really like making things on my own, and my lab has been reconstituting developmental mechanisms in cell culture as artificially as possible. For instance, this is a synthetic cell differentiation mechanism we've created a while ago. So there's one engineered cell, mammalian cell, and it divided into 2 daughter cells. So initially, 2 daughter cells have similar colors, orange colors, but eventually one daughter cell becomes red, then the other one becomes green. So this is because of the artificial gene circuit we created in this cell. I won't explain the detail, but because of this gene circuit we created, the neighboring cells can communicate with each other and then replaces each other's expression level. And it's a classic bistable system, and eventually one cell selects the red color, and then the other one becomes green. So this is reminiscent of differentiation, spontaneous cell differentiation process. That's why I would call it a synthetic cell differentiation process. And similarly, we've created a synthetic cell pattern formation. Those report a positive domains and negative domains were formed in a self-organized manner because of the gene circuit we created. So after creating a synthetic cell differentiation mechanism and cell patterning mechanism, our focus is currently on synthetic tissue deformation or synthetic morphogenesis. As you know, development means 3D tissue morphogenesis. So we wanted to manipulate tissue deformation. We wanted to make a neural tube folding, for instance. And as the actual mechanism to manipulate tissue deformation, we focused on apical constriction. So apical constriction is a mechanism by which the apical side of epithelial cell shrinks, and apical constriction can change the cell shape from rectangular to triangular. And if this apical constriction happens in multiple cells simultaneously, then this can bend a cell sheet. So we wanted to manipulate this apical constriction, and recently 3 people in the lab, Guillermo Martinez, Nuria Taberna, and Mami Takayama, created a novel optogenetic tool to induce apical constriction in mammalian cells. So this is an apical, we are looking at the apical areas of MDCK cells and epithelial cell line, and we eliminated this cell at the center. And upon elimination, the apical area of this cell shrank quickly. This tool, optogenetic tool, was termed an OptoShroom3. And OptoShroom3 is based on a gene called SHROOM3, which is a non-regulator of apical constriction. The N-terminus of SHROOM3 localizes at the apical junctions, and C-terminus of SHROOM3 can bind to ROK kinase. So therefore, the role of SHROOM3 is to recruit this ROK kinase to the apical side and then induces apical constriction. And what we did was basically to make a split version of this SHROOM3 gene. And we fused the N-terminus and C-terminus of SHROOM3 with eyelid and SSPB. And these eyelid and SSPB can bind to each other upon blue light illumination, and therefore reconstitutes the full length of SHROOM3. So that's our tool. And this OptoShroom3 can induce 25% reduction in the apical area within 1 hour. And if we eliminate multiple cells, it can induce tissue displacement. So we eliminated this area, and then upon elimination, the neighboring cells were pulled towards the center, as visualized in the PIV analysis. Those cells were pulled towards the center. So we can induce tissue displacement by using OptoShroom3. We can also induce a 3D tissue deformation. In this case, we made an MDCK cell colony on a thick, soft gel. And apical is the upper side, basal is the bottom side. And upon elimination, the apical constriction triggered the coiling or folding of this cell sheet. So we can induce tissue folding just like a neural tube folding. But actually, our main purpose of developing this OptoShroom3 was to manipulate organoids, the shape of organoids. And as a model organoid, we focused on mouse optic cup organoids. in collaboration with Mototsugu Eiraku and Nozomu Takata's group. Mouse optic cup organoids start as an aggregate of mouse ES cells, and those aggregate forms nice neuroepithelial layer, should monolayer, and this neuroepithelium invaginates eventually to form an optic vesicle, and this optic vesicle invaginates again to form a cup shape. And we focused on the vesicle stage, optic vesicle stage, this invagination stage, and then illuminated those vesicles, optic vesicles. In an optic vesicle, the inner side, inner lumen is apical and outer surface is basal. And upon illumination, optoshroom 3 caused the shrinkage of apical surface. The apical lumen shrank quickly, and individual cells became taller. So we can manipulate the shape of organoids by using optoshroom 3. Here we tested whether optoshroom 3 induces the thickening of neuroepithelia. We illuminated only this part, and upon illumination, the layer became thicker. The individual cells became taller. Yeah, so 1 hour after 1 hour illumination, the epithelia, neuroepithelia, became thicker. And we also wanted to try another organoid. And in this case, we chose a human brain organoid in collaboration with Silvia Benito and Madeline Lancaster's group. So this is the early stages of human cerebral organoids. So we need to make aggregate of human iPS cells, and then those iPS cells become neuroectodermal state. And in this stage, the inner side is basal and outer surface is apical. And after adding matrigel, the apical-basal polarity is inversed. And now apical is the inner surface and outer surface is basal. So this stage is similar to mouse optic cup organoids. So this is similar to the later stage of human brain organoids. Apical is inside and basal is outside. But we wanted to test the earlier stage where apical is the outer surface. So we illuminated this side, this part, and then upon illumination, the boundary became flattened or even invaginated a bit. As I said, apical is the outside and basal is the inside. And if apical is, in the case of optic vesicles, upon illumination, the apical lumen shrank. But if apical is the outside, then it induces the flattening or invagination of the tissue. So I think it's very interesting. Optoshroom 3 apical constriction can induce different types of tissue deformation depending on the initial tissue context. So it can induce tissue displacements in 2D, or it can induce folding or coiling of the cell sheet on a soft gel. It can induce shrinkage of lumen if apical is inside, and it can induce flattening or invagination if apical is outside. So we just posted this preprint in a bioarchive server. So if you are interested in, please let us know. Please take a look. Yeah, that's almost it. But finally, I just needed to mention that similar optogenetic tool or similar manipulation of tissues were already done by Stefano De Renzi in Drosophila. So Stefano's group developed a tool where raw GEF is translocated to the plasma membrane upon blue light illumination. And they illuminated this area, and this illumination induced a quick apical constriction and invagination of the tissue. So it's a very nice tool, and we actually created a similar raw GEF-based tools in mammalian cells. But with our hands or with our tissues, it didn't cause clear apical constriction. Probably because with this tool, we need to illuminate the apical area, apical lumen specifically. But with our Optoshroom 3 tool, we don't have to use 2-photon microscopy. We can just illuminate the entire cell. But this Shroom 3 tool can localize at the apical side from the beginning. Therefore, even with one single photon microscope, we can induce nice apical constriction. So yeah, if you are using 2-photon microscopy and if you are using Drosophila, Stefano's tool would be perfect. But if you are using vertebrates or if you don't use 2-photon microscopy, our Optoshroom 3 tool may be better. So that's it. And basically, my group tried to recapitulate or reconstitute developmental mechanisms in vitro by using stem cells. And we use those in vitro models to compare different species. And we sometimes use them for disease modeling. And also, we developed several genetic tools to manipulate organoids and in vitro models. Yeah, that's it. And the first part was done mainly by Mitsuhiro Matsuda. And Maria Costanzo and Jorge Lazaro also contribute to the project. And the second part, Optoshroom 3, was developed by Guillermo Martinez, Nuria Tabelna, and Mami Takayama. And I have several wonderful collaborators, but I think I've introduced all of them during the talk. So yeah, that's it. We are also looking for new postdocs and PhD students. And thank you for your listening.
Thank you, Miki. That was awesome. And everyone, please input your questions to the chat box, or actually you can raise your hands and ask a question yourself directly to Miki. I might kick off the question. So this difference in the timing, developmental timing in mouse versus human, and what is the mechanistic basis for it? I'm just wondering. So are the cells larger in human? Yeah, is it? Can that?
That's an interesting question. So our measurement is very preliminary, but we think human cells are larger than mouse cells. Human presomatic mesenchyme cells are larger than mouse presomatic mesenchyme cells. Probably 1.7 times or 2 times larger in the volume. And if the molecular numbers are the same between mice and human cells, then the concentration may be lower in human cells. And that could be the reason why human cells show slower biochemical reactions, but we don't have any evidence.
Do they divide slower as well?
Yeah, exactly. Yeah, cell cycle is also about 2 times slower in human cells. This could be due to slower biochemical reactions of cell cycle regulators, but again, we don't have any evidence.
Thanks. So Tom, would you like to ask directly? Maybe open up the mic, or should I read?
Hello. You are muted, maybe? I can't hear you. Tom, I can't hear you.
Something is going on because you are either muted or muted. Yeah, I see the sound.
Yeah, I think I was muted by the system. Yeah, so the oscillatory work is really interesting, especially that humans oscillate longer than rhinos was a bit of a shock to me. So if you could dream up whatever animal stem cell you would test next, what would it be and why?
Elephants. My favorite animal is always elephants because of this book of Mouse Time and Elephant Time. But I'm in general interested in very larger species like elephants or whales, or very small species like shrews. So those are my dream animals.
I was wondering whether it was related to gestation time. And
yeah.
so maybe there would be some organism, some animal with a very long gestation time.
Yeah, that's a very good question. So we initially thought body size may be correlated with biological time, but at least for the segmentation clock periods, it's more correlated with the length of organogenesis phase. So I divide the entire gestation period into two phases: organogenesis phase or morphogenesis phase and growth phase. So in human, within 60 days, the morphogenesis stage ends and then growth stage starts. And if we compare the entire gestation period, it's not so correlated with the segmentation clock periods. But if we focus on the length of this morphogenesis stage, it's well correlated with the length of segmentation clock period. So I think the segmentation clock period is a proxy for the length of morphogenesis stage.
Okay, thanks.
Actually, I'm not sure about rhinos, but at least cows show shorter morphogenesis stage than humans, so that makes sense.
Okay, cool. Thank you. And Matas, would you like to ask yourself the question? If you similar question. Well, maybe I can read. So are you planning to induce non-mammal models in your pre-somatic methods? For example, primary cultures from Annamites could provide a robust cross-species comparisons?
And ammoniotes. Yeah, I'm quite interested in other species. I'm particularly interested in snakes, for instance. Snakes show very long segmentation clocks. So in our body, we have only 40, 50 vertebrates, but snakes have more than 200 vertebrates. This means that segmentation clock continues for a very long time in snakes. And I'm quite interested in that. But the problem is we don't have ES cells nor iPS cells for snakes. That's one problem. And another issue is temperature, body temperature difference. So we focus on homothermic animals right now, and so that the comparison is easier. If we include heterothermic animals, we need to somehow compensate this temperature difference because temperature greatly affects the biochemical reaction speed. So how to normalize it, and it's a difficult part.
Could that be also a reason why those sort of the species that live in non-37 degree and ranges have a very, very diverse body plans and segmentation, numbers and things like that? But the temperature difference, do you think that could?
So I don't know, and if the temperature goes down, that generally slows down things, right? And slows down all the biochemical reactions. But still, for instance, zebrafish, they are normally cultured at 25 degrees, but they show much faster segmentation clocks. So I don't know to what extent this temperature difference can explain difference.
I see. Interesting. Okay. There's another question from Singh. Would you like to ask a question yourself? Maybe not. Then, yes, you can probably read a great talk. Want to know, have you considered epigenetics, mRNA, for the prolonged length in humans as more complex differentiation is needed?
Yeah, that's an interesting possibility. For the segmentation clock, I don't think epigenetics is so important because the time scale is quite short, and one cycle is just 2 to 3 hours in mice, and in human, still it's 5 to 6 hours. But if we think about the longer scale clocks, let's say brain development that takes more than 100 days in humans, so in that case, for those slower clocks, probably epigenetic clocks may play a role. And yeah, some people are saying that those epigenetic factors can regulate the timing of brain development, and that may be the cause of this interspecies, the slower human brain development. But for the segmentation clock, I don't think an epigenetic mechanism is so important.
Because the clock is so simple, I guess.
Simple and fast.
I have another question, combining different parts of your talk. So you have such a cool synthetic differentiation, circuits. Do you think those circuits will be slower or faster depending on? Yeah,
that's a very interesting question. I actually wanted to introduce a synthetic oscillator into human or mouse cells, and then compare the speed of synthetic oscillators. We tried a few gene circuits, but it didn't show a very nice oscillation. And so to be honest, I don't understand why Hes7, this segmentation clock, shows such a nice oscillation. We think this one factor negative feedback loop. I always say that the mechanism is one factor negative feedback loop, but the oscillation is too beautiful considering this simple mechanism. Whenever we try to make a synthetic oscillator, the oscillation is much noisier, and it's really hard to define the oscillation periods clearly. So but I'm looking. If you know a very good oscillator that shows a nice oscillation, I'd love to try. That's a very interesting possibility.
Hi, great. Anybody else? If you're not, let's close close this the seminar by thanking Miki again. And yes, if you want to meet Miki, ask more questions in a more relaxed setting, please stay on, turn the camera on, and meet Miki. The recording has stopped, and please stop the recording, Kami. And yes, right.